Hello everybody, welcome back to another recording for Biology of Cells and Tissues. So we're going to talk about these special sensory organs. In this course, typically these are grouped together. However, I went ahead and separated them out as I think it's going to be a little bit better for us to look at them individually. So we're going to start with the eye. So some of our learning objectives. We're going to look at some general information about the senses first, and then we'll talk about the anatomy and functions of the eye, as well as the histology of the different layers. So in general, we have our general senses being temperature, pressure, fine touch, pain, and then proprioception. I went ahead and included some of the specific anatomical structures that actually deal with these different ones. Some of them we've already talked about. For example, the Pacinian corpuscles and the Meissner's corpuscles we talked about when we were getting into the skin lecture. And then some of the other ones we will continue to look at, so like muscle spindles and Golgi tendon organs. You'll focus in more on physiology 1, and some of these other ones you'll take a deeper dive in neuroanatomy. The special senses, smell, sight, taste, hearing, and then balance or equilibrium. The two that we focus on in this course are sight and hearing and balance, mostly because these are the most important special senses to our functioning as humans. Vision, specifically, is one of the most important ones. It's our dominant sense. In fact, 70% of our body's sensory receptors are found in the eye, as well as half of the cerebral cortex, so half of that main chunk of the brain, is involved in visual processing. This is an extremely important component of our brain. So here is the structure of the eye in general. You have each of the different components, everything here nice, neat, and labeled. This is a really, really good way to kind of practice all of these. What I also did is I took a little bit of time to make a chart going through each of the structures pictured there and having the definition of the structure as well as the function of those structures. If you want to take a look at those, you can do so getting my notes. The link is in the description below in order to be able to gain access to all of those. So the structure of the eyeball. So we're going to start with the wall of the eye. So the wall of it all. And it is divided into 3 layers. We have a fibrous layer, a vascular layer, and then an inner layer. A lot of those are going to have subsections as well, so we'll take a look at each of those. As well, the inside of the eye, the whole structure, the entire inside, we call that the internal cavity, and that's filled with a fluid that we call humors. There's specifically 2 types of humor, aqueous and vitreous. The aqueous is in the anterior segment, so the lens right here, the lens which helps to focus the light onto the back of the eye. Anything in front of the lens is our aqueous humor, and anything to the back of it is our vitreous humor. That's very, very often a test question right there, either being what splits the anterior-posterior segment, being the lens, or which of the humor is found in which segment. Aqueous anterior, so A goes with A, V goes with P. So the fibrous layer itself, this is the outermost layer. It's very tough and protective, and it's made up of dense avascular connective tissue, and it's made up of 2 different components, the sclera and then the cornea. So the sclera, you can see, is a vast majority of the eye on the outside of the wall. It covers pretty much all of it, except for the very front, where our iris and our pupil are. That is the cornea. So the sclera is white, opaque, and the posterior region of the eye. When really it's posterior lateral, it's all of the eye except for the very front cornea. It protects and shapes the eyeball, and it is also serves as an anchor for all of the muscles of the eye. You will spend a lot of time learning about the muscles of the eye. I'm just going to do a quick intro here. SO4, LR6, all over 3. This has to do with how our muscles are innervated of the eye. They are innervated by cranial nerves. Most of them are innervated by our cranial nerve 3, which is our oculomotor nerve. Oculo, eye, motor, movement. Except for 2 of them, the SO, the superior oblique, is innervated by our cranial nerve 4, known as the trochlear nerve. And then our lateral rectus is innervated by cranial nerve 6, known as the abducens nerve. Like I said, you'll see all these muscles a bunch of times. It's kind of easy to sort them into 2 groups. We have the rectus muscles and then the oblique muscles. The rectus ones, there's one on each side. There's a superior rectus, inferior rectus, lateral rectus, medial rectus, and those, when they pull on the eye, they move the direction, they point the eye the direction that the muscle is called. And then we have a superior oblique and an inferior oblique, which are responsible for kind of depressing as well as a torsional or rotational component of the eye. So you'll see all of that, and that really gets into kind of, how the expression, the eyes are the window to the soul. The eyes are also the window to the brain. There's a lot of pathology and things that we can tell based off of looking at the eyes, the way that they move, the way that they relax, the way that they sit, how our vision is, as well as looking on the inside of the eye as well. Okay, continuing with the sclera. Posteriorly, the sclera continues where the optic nerve actually exits, right back here, and the sclera becomes continuous with the dura mater of the brain. Next, the cornea. So this is the transparent anterior 1/6 of the fibrous layer, and it forms a clear window that lets light enter, and it also bends light as it enters the eye. There's epithelium that covers the inner and the outer surfaces with something called a stroma in the middle. The stroma makes up most of the thickness of it. So the outer surface is stratified squamous non-keratinized epithelium. We always know the stratified squamous, that is our most protective layers. We want to make sure that our eyes are well protected. The inner surface is actually endothelium, so simple squamous epithelium. We say endothelium, has to do with it when it's in contact with fluids. And so the endothelium regulates water content to maintain the transparency. The stroma in the middle, not usually asked about, but it is made up of water and collagen and is really just there for the structural support. Additionally, the external cornea contains a lot of pain receptors. We know our eyes are very, very sensitive to anything that gets into them, and it hurts a lot, as well as even to wind, to breath, to movement, anything like that. So these pain receptors contribute to the blinking and the tearing reflexes. Both of those reflexes are actually controlled by our cranial nerve 5, the trigeminal nerve. You will learn eventually in, I believe it is Tri 4, your physical diagnosis course, when you start to do some of the physical examinations of patients. You'll practice this blinking reflex, the corneal reflex. You take a little cotton wisp, like a wisp of cotton ball, and you gently touch the cornea and causes a blinking reflex to test the cranial nerve 5. Here is a histological view of the cornea right here. So here is our external stratified squamous epithelium. There's our endothelial layer, and then the stroma right there in the middle. The next layer is the vascular layer. The vascular layer is also called the uvea. So either of those words work. You have the middle, this is the middle pigmented layer of the eye, and there's actually 3 regions: the choroid, the ciliary body, and then the iris. So the choroid region, you can see this is essentially the whole posterior portion of the eye. And its job is to supply blood to all of the layers of the eyeball. Remember that the outer connective tissue layer really doesn't have any vessel. It's avascular, so it doesn't have any vessels. So the choroid is what supplies all the nutrients and stuff to that outer layer. And it contains a brown pigment from melanocytes in order to absorb light and also prevent scattering, which would cause visual confusion. When you hear that word scattering, I want you to kind of think of like a disco ball. That's what it's referring to, scattering. Sometimes if light goes in and it hits things at just the right angle, it can bounce off. And so you can get light kind of going all over the place. And that would cause a lot of confusion in the brain as it tries to process all of that information. So it does a good job. It absorbs the light after it hits the retina to prevent it from going all over the place and our brain getting confused. The ciliary body, which is this portion, kind of this muscular portion right here, that actually connects to these ligaments. We call these the suspensory ligaments. That hold the lens in place. These are it's a thickened ring of tissue that surrounds the lens. It's kind of hard to see from this because this is a sideways cut or a sagittal cut. This is what it looks like from looking straight in. You have this ciliary muscle, the ciliary body muscle right here, and then all of the different zonular fibers or that suspensory ligament. So it's called a ciliary zonule or a suspensory ligament that all connects to the lens. We have ciliary muscles, which are smooth muscle bundles that adjust the shape of the lens for focusing. So when you have looking at something at a certain distance and then bring it up closer and further away, or when you jump from looking at something far away to looking at something close, it takes a very short period of time, but your eyes readjust and refocus. And that's actually this lens and these muscles either contract or relax in order to change the shape of our lens. The lens is a lot squishier than you would think and much more flexible, especially in young people. And so you're able to get more of a nice balanced shape of the lens in order to focus the light that's coming into our eyes. There we go. And there's also a structure called the ciliary process, which is a capillary-rich structure that is actually where the aqueous humor is secreted from. That's typically a nice test question as well. Usually you get a question on ciliary muscles, on this where the aqueous humor is secreted, and then you get a question on the suspensory ligaments. Finally, the iris is that colored part of the eye that's between the cornea and the lens. And the pupil, that's really it for the iris. That's its main function, is it just sits in the front. But it surrounds the pupil, which is an opening that regulates the amount of light that enters the eye. Close vision, as well as bright light, cause sphincter pupillae, or the circular muscles found within there, to contract and the pupils constrict. This is under parasympathetic control. Distant vision, looking far away, as well as dim lights, cause your pupils to dilate, and that is sympathetic control. The way to remember this is that with sympathetic system, it's fight or flight, and in order to fight or flee, you need to be able to see what's going on. So the eyes tend to dilate when you're in that fight or flight mode in order to be able to see all of the stuff that you need to see. So here are our muscles and how all of these work. And because these are involuntary, these are all smooth muscles. The lens itself, this is a transparent biconcave structure that's suspended behind the iris and that pupil that focuses light on the retina. It's avascular tissue, highly elastic, and it changes shape to adjust the focus. And it also separates that anterior and posterior segments, or the aqueous and vitreous humor. So this is what that lens looks like. The final layer of the eye is the inner layer, or the retina. The retina originates as what we call an out-pocketing of the brain. So it's very, very closely connected or linked embryologically to the brain itself. It contains millions of photoreceptors that transduce light energy. Transduce means they translate it, or they take it in and they figure out what's going on. You have neurons, and you also have ganglion cells. Specifically in this layer as well, the inner layer of the retina also is made up of two separate layers, an outer pigmented layer and then an inner neural layer. The pigmented layer is the innermost layer of the wall of the eye. It is a single cell thick lining. So one cell around the whole eye. Let's just say one cell in each spot, making a sheet of single cell thick. It extends around the interior of the eye and covering the ciliary body and the iris. It absorbs light, prevents it scattering. It also does phagocytosis of photoreceptor cell fragments. So when our photoreceptors start to break down or have any damage to them, this pigmented layer cleans it all up. They also store vitamin A, which is crucial for the synthesis of something called retinal. This is required for the photoreceptors to work to make the pigment rhodopsin. This will come back when you start talking more about neuroanatomy and neurological function and stuff of the eyes. For now, things that I want you to remember: it stores the vitamin A. Vitamin A is needed for retinal. I say it like that, retinal, because there's also another one called retinol with an OL. We want to remember retinol is the one associated with the eye. And then you might want to remember the word rhodopsin. Link those three things together: vitamin A, retinal, rhodopsin. That will get you through testing here, and those are going to come back significantly, not just in more courses, but also in boards. Biochemistry talks about a lot of these things, as well as our physiology stuff. The neural layer. This is the transparent layer that runs anteriorly up to the ciliary body. And the anterior end of the retina has some serrated edges. So it's these serrated edges right here, that ora serrata. That is where the retina ends. So it runs all the way along the back and stops right there and gives that serrated edge appearance. There are three types of neurons there. There are photoreceptors, which work on detecting the light and convert it to neural signals. Bipolar cells, they are intermediaries. They transmit the signal. And then ganglion cells, which collect all the signals and then carry them to the brain through the optic nerve. Here's the microscopic anatomy. So we have our, right here, this is the retina out here. This is the single pigmented layer. We have our photoreceptors, the rods and the cones, which we'll talk about in a little bit. We have the bipolar cells, which connect them to the ganglion cells. And then all the ganglion cells all run together into turn into the optic nerve. Cranial nerve 2 with the optic nerve. Here's the microscopic anatomy. This is what it looks like. We have the choroid, pigmented layer, rods and cones. This whole layer right here is the rods and cone layer. And then the bipolar cells, and then those ganglion cells. The retina also contains a structure called the optic disc. This is where, you can see right here, this is actually where the nerve exits the cell. It's also called the blind spot because the nerve exits this eye there, and doesn't have a layer of the photoreceptor cells, we don't get any information from that one specific spot. So that's why it is called the blind spot, because we literally cannot see there, because any light that's entering and hitting that specific spot does not register as anything, because there's nothing there to pick it up. And there are two types of photoreceptors. We have our rods. It looks like a rod, and a cone. It literally looks more like a cone shape. I put a lot of information in here. I'm going to point out the very specifics that I want you to know. So rods are highly sensitive to light, so they're really good for the dim light or night vision. They are more in the periphery of the retina. They cannot detect color. They're more for our black and white style vision, grayscale vision. That's why our grayscale, that's why at nighttime things tend to look more gray than anything. As well, there's no sharp image production. So things tend to be a little bit blurrier at night. Cones are the opposite side. These are for our bright light or the well-lit conditions. You get higher resolution as well as color vision. Specifically, we're talking about, there's an area called the macula lutea. This is at the posterior side, near the posterior pole, near the very back of the eye, and it's just next to the blind spot. And this is where we have a high density of cones, right there, the macula. This is also something that you're going to look at. I should have thrown a picture in here, but this is something that you're going to look at specifically when you are looking at diseases potentially of the eye, when you're doing that examination of the eye. Within the macula itself, there's a tiny pit that's in the center that only contains cones. I should say that only contains cones. So there's only cones in this one specific spot. So it is the region with the best visual acuity. This is actually our focus point. This is where we are predominantly focusing light. So you can see things out of your peripheral vision, but the thing that you're looking straight at and focusing on, our eyes, they move and they focus in order to actually center the light directly onto the fovea centralis. And really, that in general is how our eyes all kind of work. Everything works together. So the muscles behind of our iris are going to dilate or constrict, make sure that the pupil is the correct size as light comes in. Our ciliary muscles are going to stretch or contract or relax and stretch the lens to the correct thickness in order to focus the light onto this region of the retina, specifically mostly the macula, in order to allow us to focus on something. The light hits the layers of the retina, and it gets read and converted into a signal by our photoreceptors. That message gets passed through bipolar cells into the ganglion cells, and those ganglion cells continue and carry that information through the optic nerve all the way to the brain, and then the brain processes it. So that is really all you need to know at this point about the eye. You're going to learn a lot more about it in the future. I added this as well just to start you on your journey of seeing a couple of common terms when it comes to pathology of the eye. So terms like papillary edema, papilledema, papilledema, I believe that's how it's spelled. There we go. Papilledema, when you get swelling of the optic disc, diabetic retinopathy, macular degeneration, glaucoma, hypertensive retinopathy. These are some conditions that I just want you to see the words now, because these are going to be some big ones that you're going to talk about in future courses. I also included some causes, some of the specific findings. If you want to take a look at those, these are not tested in this specific course. However, in the future, these 100% are going to come back, and these are the big ones that you guys are going to talk about when it comes to eyes. Okay, and that is our eye lecture. Of course, if you can, leave a like on the video, subscribe if you haven't already, and I will see you in the next one. Have a wonderful day, and good luck studying.